This lecture is entitled The Transport of Oxygen, Carbon Dioxide, and Blood in Body Tissues. So far we have talked about the means by which we get oxygen into our lungs. We talked about pulmonary ventilation, the physical act of getting air into our lungs. We looked at the principles of gas exchange, and now we're going to look at how the oxygen, CO2 are transported in our blood and within our body tissues. You'll notice in your notes, the notes are pretty fairly complete. There's only a couple spots that you would have to fill in some notes. So what I'm going to be doing, just keep in mind that I will probably be staying within a picture for a very long period of time, but what I will be saying will be written for your notes in order. So you can look at the picture on the screen and kind of follow the notes as I go along. But I first want to do is kind of an overview. Is what we're going to be talking about is the different ways in which we transport oxygen and CO2 in our blood. The oxygen is actually transported 2 different ways. Carbon dioxide is transported 3 different ways. The reason why that we have more than one means of transporting these gases is that both oxygen and carbon dioxide have limited solubilities in blood plasma. So one of the ways in which they're transported is dissolved in plasma, but we produce more carbon dioxide than the plasma can absorb and transport. We need a lot more oxygen that can be transported, again, dissolved in plasma. So we solve the problem by using red blood cells. Now, red blood cells, one of the things they'll do is they'll carry hemoglobin. Red blood cells can transport oxygen bound to hemoglobin. As well, the red blood cell can transport carbon dioxide. Now, carbon dioxide is, this picture doesn't have it, but CO2 is also physically dissolved in plasma, but the red blood cell can bind to carbon dioxide directly, but it also can convert some of that carbon dioxide to bicarbonate. So we're going to be looking at those later. So we have multiple ways by which we transport these gases. Big thing to keep in mind is when we look at oxygen, we're going to see this picture here is the alveoli. It's going to go from the alveoli into the blood. And then at the level of the tissues, it's going to go from the blood to the tissue. The carbon dioxide is going to go in the opposite direction. So we're going to be producing it in the tissues, transporting it in the blood somehow, one of the 3 different ways, and then at the level of the lungs, we're going to, CO2 is going to go out into the alveoli, and then we're going to exhale it. When I talk about oxygen and CO2 in regards to partial pressures, if I use that term partial pressure of carbon dioxide or the partial pressure of oxygen, I am only referring to the gases that are dissolved in plasma. Only gases dissolved in plasma exert a partial pressure. If it's bound to hemoglobin, it no longer exerts a partial pressure. So we're going to be, when we go through all the partial pressures in different locations in our body, I'm only referring to that particular gas that's in the dissolved state. Now, one thing that's in your notes, I'll come back to this picture in a moment, that's not in your notes that you need to be aware of and take and write it down right now, is that there are abbreviations that we'll use for partial pressures, and you're required to know that abbreviation. You can't ask Dr. Kayes during the middle of the exam, well, what is that supposed to mean? You're supposed to know it. The capital P is referred to as the partial pressure. If I use a capital A next to it, sometimes I'll put it in a subscript, but it'll be a capital A, I'm referring to that particular gas in the alveoli. So if I say I have P capital A O2, I'm asking for the partial pressure of oxygen within the alveoli. If I use a little a lowercase a, that is the partial pressure of the gas in the arterial blood. If we use the lowercase v, that refers to the partial pressure of that particular gas in venous blood. We do not have abbreviation for partial pressures of gases in the interstitial fluid or the cell, but we do have these abbreviations for alveolar, arterial, and venous blood. So that is something that you may see me write on an exam instead of actually saying it out. What's the partial pressure of oxygen in arterial blood? I may use the abbreviation. Now, what I want to do is look at oxygen, specifically oxygen in the dissolved state, and look at the partial pressures of it through different parts of the circulation. So you have this picture in your notes, plus it should be, it's written out for you in word form too. So we're going to look at the various partial pressures. So we're going to start up in the alveoli because that's where we're going to pick it up. So you breathe in. Oxygen in the alveoli, the partial pressure of oxygen is 104 millimeters of mercury. Now you notice I didn't put the units throughout here, but it's all in millimeters of mercury. The oxygen will go from the alveoli into the blood because there will be a pressure gradient. The oxygen in the blood that's going up to the alveoli is only going to have a partial pressure of 40 millimeters of mercury. It's what you have in venous blood. So the initial gradient for what they call the AVO2 difference, the difference in between arterial and venous blood, is 64 millimeters of mercury. So you're going to have really quick, rapid exchange right there in the alveoli. As soon as that blood goes past the alveoli, that blood will have a partial pressure of oxygen of 104 millimeters of mercury. It will fully equilibrate. Now that blood will go to the heart, and it'll still be 104 millimeters of mercury until you get into the heart, because it's going to empty into the left atrium, fully saturated, but it's going to mix with some blood from the bronchial circulation. So about 98% of the blood in the left atrium is from the pulmonary circulation. 2% is from the bronchial circulation, which is relatively deoxygenated. So that partial pressure of that blood is only about 40 millimeters of mercury. So what's going to happen is, because of this venous admixture, ADMIXTURE, the partial pressure of oxygen in the blood leaving the heart is going to be less. And in here, you have it written as 95. It's anywhere from 95 to 100 millimeters of mercury. So it's slightly less than the blood that you saw up in the pulmonary capillaries. Now, so the partial pressure of oxygen arterial blood is anywhere from 95 to 100 millimeters of mercury. Nothing's going to happen to it yet because you can't exchange it till you get down to the level of the capillaries. And at the capillaries, oxygen is going to go from the blood into the interstitium because, again, there is a pressure gradient. As long as we have a pressure gradient, you're going to have the movement of oxygen from the capillaries into the interstitium. Now, the partial pressure of oxygen in the interstitium is about 40 millimeters of mercury. So you have a gradient of anywhere from 55 to 60 millimeters of mercury here, so that's going to allow for pretty quick exchange or diffusion. And then the oxygen is then going to go into the cells, because that's where we're going to use the oxygen at the level of the cell. Now in the picture, we have the partial pressure of oxygen in the cell of being 23 millimeters of mercury. Now that can range, that's an average number. That can range anywhere from 5 up to 40 millimeters of mercury, depending upon the metabolism of that cell, how far we're away from the blood source, so that we're just giving you an average. But obviously it's going to be less than the interstitium, because this is where we're going to use it to use for electron transport and other things. So the oxygen will continue to diffuse from the interstitial space into the cell, again, because there is a gradient. The oxygen will be utilized by that cell and will produce CO2 as a byproduct. And we'll look at CO2 transport a little bit later, but now we're still talking about oxygen transport and oxygen levels in different areas of the circulation. So what happens is, by the time this blood has gone by the interstitium in the capillaries into the cells, all this oxygen is going to diffuse into the interstitium in the cells, and by the time it leaves, that partial pressure of oxygen in the capillaries is going to be fully equilibrated. What you have in the interstitium will be the same. It gets to where there's no more gradient. And so the venous blood, the partial pressure of oxygen in the venous blood is 40 millimeters of mercury. And this is what goes back into the right side of the heart, up eventually again back up into the lungs, where we're going to pick up oxygen again. So you'll notice again, so the values you need to remember: the partial pressure of oxygen in the alveoli, 104. In arterial blood, anywhere from 95 to 100 millimeters of mercury. The interstitium and the venous blood is the same. Okay, so it's 40 millimeters of mercury. And in the tissue, or the cell, it ranges anywhere from 5 to 40. The average is about 23 millimeters of mercury. Minimally, you should be able to tell me, just by using logic, put the partial pressures of the gases in order from highest to lowest. It's going to be the highest in the alveoli because we're getting brand new oxygen. It's going to be lowest in the cells because they're utilizing the oxygen. And everybody is going to be somewhere in between. And as long as you have a gradient, oxygen is going to diffuse from point A to point B. If there's no gradient, you'll have no diffusion. The one thing to keep in mind, though, if we go back up here to the alveoli, that diffusion of oxygen, the rate can change if metabolism changes. Say you're exercising. Your need for oxygen is going to go up. So we're going to need to deliver a lot more oxygen. So there's a lot of things that's going to take place to allow us to deliver more oxygen to your tissues. You're going to have an increased amount or rate of blood flow in the lungs. We're going to increase the diffusing capacity for oxygen by opening up unperfused capillaries. We're going to have a more ideal ventilation perfusion ratio in the lungs. And also what's going to be happening, we'll be seeing later, is looking at what happens to the oxygen hemoglobin dissociation curve. So, but big thing is during exercise, usually, if you look at normal individuals, the limiting factor for us to get enough oxygen in our body is not at the level of the lungs. We are well adapted to be able to get a lot of oxygen across that respiratory membrane into the blood. The limiting factor is our ability to get the oxygen to the level of the tissues. How well are those leg muscles, the arm muscles, how well the blood vessels serve those. It's not about getting oxygen in the level of the lungs. Now, so we've covered the partial pressure of oxygen in the different areas of the circulation. Now we've got to look at carbon dioxide. So we're going to start where the CO2 is produced. So we had started with oxygen, where it came in was at the alveoli. So we're actually going to start carbon dioxide at the level of the cells because that's where it's being produced. So you get that from the reaction which pyruvate is converted to acetyl-CoA. You get that in some reactions of the TCA cycle. So you're always producing carbon dioxide. Now, carbon dioxide diffusion is always going to be opposite that of oxygen. And the only exception is that CO2 diffuses much faster because carbon dioxide is much more soluble. And because it's much more soluble, that the carbon dioxide requires a lower pressure difference. So you're going to notice the difference in the partial pressures of carbon dioxide in the different parts of the circulation is not very dramatic. So I always find it harder to remember the partial pressures of CO2 in the different parts of the circulation because they're so close to each other. The partial pressures we have are listed for you in your manual because it's going to give you ranges. And this here on the picture doesn't give you the range, but there's range. As long as you have a very slight difference, you will get diffusion. Minimally, for carbon dioxide, the partial pressures of carbon dioxide, just like I did with oxygen, you should know where it's the highest and then eventually who's the lowest. If you know the orders, like I need to go from the cell to the interstitial fluid to the capillary blood to the venous system, up to the lungs, arterial blood, back down to the level of the tissue, you know the order is always that's going to be the highest CO2 down to the lowest CO2. For your exam, I absolutely require you to know the partial pressure of CO2 in the alveoli and arterial and venous blood, because those are things that come up a lot. Boards may ask you the partial pressure of CO2 in the interstitium as well as the cell. So you should be review those before you take the boards, but for your exam I will not ask you those two. For oxygen, I minimally want you to know the partial pressures of oxygen, at least for this thing, it's the alveoli. You need to know it in arterial blood, and you definitely need to know it in venous blood. So again, you don't have to recognize the interstitial partial pressure of oxygen or the cell, but you may be required to know that for boards. But I still may ask you partial pressure of oxygen of the interstitium. Is it greater or less than arterial blood or something like that? You should be able to tell me what is it in comparison and not necessarily the actual number. So let's go over the partial pressures of CO2. So at the level of the cell, it's going to be the highest because that's where you're producing it. So it ranges anywhere from 46 to 50 millimeters of mercury. In order for CO2 to diffuse into the interstitium, the partial pressure of CO2 in the interstitium has to be less. And it ranges anywhere from 45 to 46 millimeters of mercury. Well, why is it going to be a little bit lower? Because CO2 is always diffusing into the capillaries, because eventually you've got to get rid of it. So in the capillaries, the arterial blood that's bringing the fresh blood down, the partial pressure of CO2 in arterial blood is 40 millimeters of mercury, so you're going to have a gradient. So it's going to go easily into the capillary blood. That's going to eventually become venous blood. So you see it's the 45 millimeters of mercury, because it's picking up some more CO2 from at the tissue level. When the carbon dioxide goes back up to the heart, back up to the lungs, up in the alveoli, it's 40 millimeters of mercury, and so CO2 is going to go from the blood out to the alveoli because there's a gradient. There's a 5 millimeter mercury pressure gradient. So CO2 is going to go from the venous blood out to the alveoli. So as long as there's a gradient, you're going to get diffusion. So the values that I want you to know for the partial pressures of CO2 and oxygen, you need to know it in the alveoli, you need to know it in arterial blood, you need to know the values in venous blood. Even though you don't need to know the actual value for the interstitium or the cell, you should know how it relates to the others. You also, though, have to remember the partial pressures of CO2 and oxygen in the tracheobronchial tree. What is it, or what's it in the atmosphere? What's it in humidified air? That too. So you have to remember that from the previous lecture. Now what we're going to do next, we just reviewed the partial pressures of the gases in major locations of your body. We need to look at how we transport specifically these gases in other forms. So we're going to look at the transport of oxygen first. Oxygen is transported two different ways. Only 3% is actually found dissolved in plasma. Very small amount is actually dissolved in plasma. Most of the oxygen is transported in your blood bound to hemoglobin. So knowing the fact that most of your oxygen is bound to hemoglobin, if I have problems with insufficient amount of hemoglobin, I will definitely have issues with ischemia, because not ischemia, hypoxia, because I'm not going to be able to have and be able to carry sufficient amounts of oxygen since most of it's bound to hemoglobin. But what I want to do is review with you the structure of hemoglobin, which was covered in biochemistry. So hemoglobin, the hemoglobin molecule in adults, has 4 subunits, 2 alphas, 2 betas. Each globin chain has a heme ring in the center, and in that heme ring, you have this iron, and oxygen actually binds to the iron within the heme ring. The globin chains are just there to prevent water from coming here, which would oxidize the iron. It also prevents, if oxygen's here, forming bridges between another oxygen that's over here, which then it wouldn't be very good for letting go, the oxygen. So the globin there is kind of a protective thing. The oxygen actually binds to the iron in the heme ring. So hemoglobin can carry how many molecules of oxygen? It can carry up to 4 molecules of oxygen. Hemoglobin is how we transport the majority of our oxygen in the blood. Remember, myoglobin is found in muscle. It would be like one of these, one globin chain with a heme ring in the center. It can only hold on to 1 molecule of oxygen, and it holds on extremely tightly. It's more for storage rather than transport. So what makes hemoglobin such an ideal candidate for transport of oxygen? So let's look at the curve, this oxygen hemoglobin curve. The curve, what it does is it shows you a relationship between the partial pressure of oxygen, so again, that's the oxygen found in the dissolved state, and how hemoglobin is saturated with oxygen. So you'll notice the shape of the curve. This curve is sigmoidal, which in biochemistry you should have looked at the myoglobin curve and the hemoglobin curve. The myoglobin curve is a parabolic curve. Not parabolic, sorry, it's hyperbolic. It kind of runs up like this. This one is more sigmoidal. So if you notice at the partial pressure of oxygen up in the lungs, which is about 104 millimeters of mercury, the hemoglobin molecule is, by this picture, looks like it's 100% saturated, but it really is not. Most of us have different types of hemoglobin, and because of the large variance of hemoglobin out there, it's mainly usually about the level of lungs about 97% saturated. And that value is going to come into play later, okay? But it's pretty well saturated at the partial pressures you get the lungs, which is what we want. Blood's going to go up the lungs. Some of the oxygen is going to be dissolved in plasma. Then some of it's going to, a lot of it's going to transfuse into the blood cell and bind itself to hemoglobin. Now if you look down here, as we move down to lower partial pressures of oxygen, which we start getting at the level of the tissues, you'll notice like about right here, the level of oxygen that you get at the level of the tissues is about 40 millimeters of mercury. Hemoglobin is still very well saturated. It's about 75% saturated in venous blood, because once the blood makes its way down to the tissue, gives up its oxygen, moves on its merry way, venous blood is about 75% saturated, and it has about 40 millimeters of mercury of oxygen. So if I say it's 75% saturated, it means the hemoglobin gave up 1 molecule of oxygen. So it still has 3 bound to it. Now, if you keep moving down to lower partial pressure oxygen, it becomes a lot steeper. So when it starts to get real low, say if you're working out really intensely, very metabolically active cell or tissue, the hemoglobin molecule will unload a lot more oxygen if it needs to. But up here, it doesn't need, for most tissues, doesn't need to give up a lot. So up here, it's not giving up as much, but if it needs to, it will unload a lot. Now, if we look at this curve going this way, low levels of oxygen, okay, it's hard to pick it up. As it goes up, it picks it up fairly easy. So very low difference or small difference in partial pressure, the hemoglobin molecule will pick up oxygen very readily. So this is why I want to talk about it this way. You may have learned in biochemistry about the concept of positive cooperativity. The way the hemoglobin molecule is designed is that if one oxygen binds to the hemoglobin, it changes the configuration. You get a shape change. It's an allosteric protein, and it changes the shape of the other globin chains, making it much easier for subsequent oxygen to bind, which is a very good thing. Get a second one on, makes it even easier for the third one to go on. When the third one binds, it makes it even easier for the fourth one to go on. Okay? But also you can think of the idea of hemoglobin being that allosteric protein in that when certain things we'll be talking about later, certain things can bind to the globin chains, nowhere near where oxygen binds to. You can get shape changes where then it may alter the affinity of oxygen for hemoglobin, which may be important in some conditions such as exercise, which we're going to be talking about a little bit later. But let's just look at this curve and look at this thing here. It says oxygen content or volume percent. That the curve can be expressed in terms of what we call volume percent. It's like an older term that they used to use. The average amount of hemoglobin that adults have is we have about 15 grams of hemoglobin for every deciliter of blood, and a deciliter is 100 mils. If I tell you that for every gram of hemoglobin that we have, we can carry 1.34 mils of oxygen, we can. So if every gram of hemoglobin can carry 1.34 mils of oxygen, if I have 15 grams of hemoglobin for every deciliter of my blood, I can carry 20.1 mils of oxygen per deciliter of blood. Now that's assuming 100% saturation. And if you have that approximately about 20 mils of oxygen at 100% saturation, this is where you see up here, this 20 volume percent. That 20 volume percent is also referred as 20 mils per deciliter. That's where that term comes into play. But earlier I told you that in reality, we are not 100% saturated at the level of lungs. It's mainly about 97%. So if I say, okay, assuming I have 15 grams of hemoglobin for every deciliter of blood, if I have 100% saturation, I can carry about 20.1 mils of oxygen. If it's 97% saturated, all you have to do is take that 20.1, multiply it by 0.97, and that will give you the actual amount of oxygen that you're transporting, because you have to take into consideration your saturation. So in your notes, you say under normal conditions, in arterial blood, we're carrying about 19.4 mils of oxygen for every 100 mils of blood. Okay. Well, what happens here? If you look at this picture, at the level of the venous blood, said it was 75% saturated. You'll notice we're over here, we're at 15 volume percent. Where that comes from, it's not quite, but it's close to 15%. You have 75% saturated, so you take 0.75 and you multiply it by that 20.1 and you get 14.4. So it's close to that 15. So what has happened is that you pretty much have unloaded about 5 mils of oxygen for every deciliter of blood at the level of the tissue. So that's like normal conditions, okay? So what happens if I need to exercise? What's going to happen with the amount of oxygen I'm going to unload from the hemoglobin molecule? Well, during moderate to severe exercise, I want to unload some more. What can happen is that the partial pressure of oxygen can actually go to as low as maybe 15 millimeters of mercury. So instead of being up here, we're going to be way down here. We're going to be a lot less percent. And so we can get down to minimum about 4.4 mils of oxygen per deciliter of blood in venous blood if the metabolic demands of the tissues are great enough. And so you've unloaded 3 times the amount that you did under normal conditions. So under normal conditions, we've released about 5 mils. Here we can release about 15 mils. So the hemoglobin molecule, if it needs to, can unload a lot more if it's necessary, which is a very, very good thing. Also consider that if I'm exercising, I'm going to need more oxygen, level the tissues. I want the hemoglobin molecule to release more oxygen. My cardiac output's going to go up to be able to deliver that blood to the tissue. So technically, if I have 3 times the amount of oxygen delivered to the tissues, and my cardiac output maybe goes up to 6 or 7 times, I can get about 20-something fold increase in oxygen transport to tissues, which is pretty good. So you can see that about the elite athletes when you start getting cardiac outputs to 6 or 7 times the normal. The hemoglobin molecule is very advantageous in a number of aspects. In your notes it says the role of hemoglobin as a quote-unquote buffer of tissue oxygen levels. So when we talk about as a buffer in this case, we're not referring to as a chemical buffer, but hemoglobin is a chemical buffer when you talk about pH balance. But any buffer, the purpose of a buffer is to prevent drastic changes in stuff. In this case, hemoglobin is a very good buffer in preventing dramatic changes in tissue oxygen levels. So if, for example, if you look up here, the level of the lungs, that's assuming my partial pressure of oxygen is 104 millimeters of mercury. The alveolar oxygen levels can vary pretty greatly depending on what's going on. It can go down to as low as 60 in some cases, or it can be as high as 500. But what's going to happen is the partial pressure of oxygen in peripheral tissues don't change as dramatically. So here, for example, I can enter an area of compressed air, very pressurized chamber in which the partial pressure of oxygen is much, much higher. But we have the hemoglobin molecule can only hold so much. You cannot bring it that much up at all. If I'm in I'm ascending in an airplane or I'm ascending a mountain, the partial pressure of oxygen, and I'm talking about a non-pressurized plane, can decrease, but the alveolar partial pressure oxygen can get as low as about 60 millimeters of mercury, and you're still going to be pretty well saturated. It'll be about 89% or so saturated, which are here 60, sorry. It'll still be about 89% saturated, which isn't bad. The tissues are still going to remove about 5 mils, and if that happens, the partial pressure of oxygen will fall to about maybe 35 millimeters of mercury, and that's not much less than 40. And so the big idea is the hemoglobin molecule can, by just the way it is, can prevent the tissue oxygen levels from varying too much. So it can deliver a lot more oxygen if it needs to the tissue, so the tissue levels don't change that much. And then we had said, like, even if the alveolar partial pressure of oxygen changes a good amount, the hemoglobin molecule, as long as it stays relatively saturated, nothing else bad stuff happens, it can deliver sufficient amount of oxygen to the tissues. What we're going to look at is hemoglobin under normal physiologic conditions, because how many of us actually go climb huge high mountains? But it happens. But what about exercise? What's the role of hemoglobin during exercise so it maintains adequate tissue amounts of oxygen? Well, what can happen, as you see here, there can be a shift in this curve. the configuration of the hemoglobin molecule can change, which alters its affinity. So if you just look at this picture, this one here up at the top represents the normal hemoglobin molecule. Under normal conditions, I should say, it's still going to be a normal hemoglobin molecule. But under certain conditions, there can be a shape change in the hemoglobin molecule. So if we look at the curve, in this case, it's been shifted to the right. So what does it mean? For the same partial pressure of oxygen, so we say 40, with a change in shape of the hemoglobin molecule, it is less saturated. So what does it mean? That means it has less oxygen molecules bound to it. Its affinity for oxygen is decreased. You say, well, some people may think that's a bad thing. No, the level of the tissue, if the tissue needs oxygen, hemoglobin's going to unload it. That's its job. It can go back and get some more later. So what type of things can cause the hemoglobin molecule to unload more oxygen and it was due to some sort of shape change within the hemoglobin molecule. So exercise is the number one, greatest example for this. So during exercise, carbon dioxide levels go up. Hydrogen ion concentrations go up because you're producing more lactic acid. You can get some hydrogen ions as a result from that. The temperature of the tissue, the muscle, increases. These things, all of them can affect the configuration of the hemoglobin molecule. And I'm going to come back to this one in a moment. So a lot of these things can affect what the shape is. Carbon dioxide, we're going to be talking about how CO2 is transported in blood, and one of the things it does is it's physically bound to the hemoglobin molecule. And it binds somewhere other than where oxygen binds to. Hydrogen ions can bind. Temperature can affect the configuration. So if something binds or the temperature is affected here, the shape of the molecule changes. And what happens is these things would cause a shift in the curve to the right, which means it's able to deliver more oxygen to the tissues. For a given partial pressure of oxygen, the hemoglobin molecule is less saturated. The affinity for oxygen has been decreased. Okay? There's a term for the effects of hydrogen ions and CO2 on oxygen transport, and they use the term the Bohr effect. So the Bohr effect is specifically refers to the effects of carbon dioxide and hydrogen ions on the oxygen hemoglobin curve. Now the Bohr effect is explained in more than one way. Decreased levels of these things or increased levels causes a configuration change in the hemoglobin molecule. So the levels of the tissues, you look down here, the levels of the tissues, these things are going to be elevated because you're working out or you're metabolically active. The curve shifts to the right, and what's that do? It releases more oxygen at the level of the tissues. At the level of the lungs, the curve shifts to the left. You're going to have increased oxygen binding in the lungs because that's what you want. You want to be able to pick up oxygen in the lungs, delivers it to the tissues. So why does it shift to the left, the level of lungs? Well, I wonder why. The level of lungs, CO2 is going to be let go. And we're going to be looking at what happens to hydrogen ions because it's all intricately related to how CO2 is transported. So the Bohr effect shows us a relationship between carbon dioxide and hydrogen ions on the ability to transport oxygen, so on oxygen transport. So during exercise, these things are elevated. So what about this 2,3-BPG? 2,3-bisphosphoglycerate. That's what the biochemists refer to it as. Physiologists like to say 2,3-DPG, diphosphoglycerate. So biochemists like to use the prefix bi, physiologists di. I'm more a physiologist, but I like BPG better. You may have heard of this compound. This is produced from a glycolytic intermediate, 1,3-BPG. You just move the location of the phosphate from carbon 1 to carbon 2. So this is a reaction that takes place in red blood cells. This product is a very important regulator of the transport of oxygen in hemoglobin. The more the 2,3-BPG being produced by red blood cells, it causes a shift of the curve to the right, which means it's going to release more oxygen at the levels of the tissues. Where you may see more dramatic effects or increases in the levels of this would be in response of going to high altitudes. One of the things that happens when we go to higher altitudes is we produce more of this product and allows us to deliver more oxygen to the tissues. It also may be a product of or increased in response to certain hormones like growth hormone, testosterone. So growth hormone, you think growth, you need oxygen to be able to grow. And so this is increased when growth hormone levels go up. Testosterone is another one that allows you to produce a little bit more 2,3-BPG and causes more oxygen to be delivered to the tissues. Now, the cells themselves don't really, believe it or not, need a heck of a lot of oxygen, considering. So it says here we need like about 1 to 3 millimeters of mercury of oxygen for resting metabolism. And the way our bodies have been designed is most cases oxygen is not a limiting factor, usually. Usually what limits or affects things is ADP, the concentration of ADP, which you probably learned in biochemistry, looking at how the electron transport chain is regulated. As ADP levels went up, the rate of electron transport increased because you need to make more ATP. Most cases, we deliver enough oxygen to the tissues, so it's not a limiting factor. But that said, we have to give you examples of where it can become a limiting factor. Normally, distance is not an issue. These cells are usually very close to capillaries, but every once in a while we have things that are not as close to the tissues, and where the distance becomes a limiting factor. It's going to be a lot farther for the oxygen molecule to go from the capillaries to the tissues. So in this case, we're going to refer to as being diffusion limited flow. That's if oxygen levels fall below the level that the tissue needs to function. That may, play a role in, say, you have a fast-growing, say, cancer cell. Fast-growing cancer cells kind of move, they grow too fast that they're going to have to rely heavily on glycolysis because they can't get a lot of oxygen for aerobic metabolism. Eventually, if those cancer cells develop their own blood supply, then everything's going to really take off too, because then they can get a lot more oxygen and grow even bigger. Now, another thing that may influence or make it that oxygen is a limiting factor is if you have a problem with flow. So instead of saying flow limited, I'm referred to it as change it to perfusion limited. P-E-R-F-U-S-I-O-N. Perfusion limited. This is where if the blood flow, my perfusion of the tissues, is a problem, I'm not going to be able to get enough oxygen for a certain period of time because the flow rate is not enough. So if blood flow rate is decreased, again, below the levels that the tissue needs to function properly, referred to as being perfusion limited flow. Best case example, hey, you got a little blockage in the blood vessel in the coronary arteries. Not get enough oxygen, not enough blood flow to that tissue, definitely oxygen becomes a limiting factor because I can't get enough blood flow there. Now, so far, we've talked about the oxygen transported by hemoglobin, 97% of it, and you learn how to calculate, and we'll do some practice where if I said how much oxygen is being transported in this patient's or person's blood, you first know how to figure out how much is bound to hemoglobin. So you take, if you know the amount of hemoglobin that that person has, you multiply that by 1.34, and you also need to know the saturation, and you multiply it by whatever percent you have. That's the majority of the oxygen that's being transported. But the total oxygen content of a person's blood is what's bound to hemoglobin, but also what is physically dissolved in plasma, that which exerts the partial pressure. So you have in your notes how to calculate oxygen transported in the dissolved state. So this is the conversion factor. If you know the partial pressure of oxygen, you multiply by this value, and you'll get the mils of oxygen per deciliter of blood. That would be the units. Mils of oxygen per deciliter of blood. It's not a lot, but it's still a component of the total oxygen content of the blood. So for example, if partial pressure of oxygen is 100 millimeters of mercury, 100 times 0.003 is 0.3 mils of oxygen per deciliter of blood. So that would be the amount. Now, during real severe exercise, the amount of oxygen dissolved can go down a lot because you're going to be using a lot of that oxygen. But hopefully, we can replace it when the exercise is finished. And one last thing I want to talk about in regards to oxygen before we take a break and then look at the transport of carbon dioxide is the effects of something such as carbon monoxide on oxygen transport. A lot of you may have heard of carbon monoxide. Carbon monoxide is an electron transport chain inhibitor, so that's going to mess with you. But what also it does is it can bind to the heme ring on hemoglobin. It binds to the same exact spot as oxygen. So it will take up a spot for oxygen. And the thing is, it binds to it a lot stronger than oxygen. It will eventually let go, but it doesn't let go easily. So what happens is the patient becomes hypoxic because the hemoglobin is bound to carbon monoxide instead of oxygen. And again, most of the oxygen that we have in our bodies is transported bound to hemoglobin. That dissolved in plasma is not enough. Now, where does carbon monoxide come from? It is a product of incomplete combustion of fossil fuels. So if you own a home or live in a home that uses gas, you have a gas heater, anything like that, if you have a purely electric home, you don't have to worry about carbon monoxide poisoning. But you get them from gas heaters. You also get it, like you hear about people that stay in their car, keep their car closed, they're in a closed garage. You get that from the exhaust fumes from the car. The carbon monoxide, fairly small amounts of it can be lethal. So this kills a number of people each year, and that's why it's so important to have carbon monoxide detectors in your home, because you can't smell it. It's a colorless, odorless gas, and you're not aware of it. Some people go to sleep and don't realize they're being poisoned with carbon monoxide. The one thing to keep in mind about if someone has carbon monoxide poisoning is if you measure the partial pressure of oxygen in their arterial blood, the levels will be perfectly normal. And the thing is, that prevents the normal feedback mechanisms in our body. It says, hey, oxygen content's low, and they have no real physical signs, outward signs of having hypoxemia, like cyanosis, because that comes from the oxygen in the dissolved state. So everything's like the mucous membranes, everything's nice and cherry red. They're not bluish color. But they're going to have outward symptoms of something's up because they're not getting enough oxygen to the tissues that need it. So what happens is, if carbon monoxide poisoning is suspected, is the treatment for it is obviously remove them from the source of the poisoning, and they will administer them like about 95% oxygen, 5% CO2 mix. The reason why they're going to give a high percentage of oxygen is they're going to try to force more oxygen in the dissolved state to force the carbon monoxide off of the hemoglobin molecule. And why they put CO2 in there, CO2 is a very potent stimulator for respiration. So it's a lot more important stimulator for respiration than low oxygen. So CO2 levels really stimulate that patient to try to increase their breathing. So obviously if they're real bad, they may not be breathing all that well, but that mix promotes them to breathe in that mixture a little bit better. There's other treatments that they use to try to get rid of the carbon monoxide, but this is just one of the basic things that they do. What we're going to do now is just take a break, and when we get back, we'll talk about the transport of carbon dioxide, which won't take very long.